This is the third part. We're going to be taking a look at the function of the plasma membrane. And with this function, really what we're going to be doing is focusing on the different ways substances can cross this biological membrane. When we do that, one of the things that we do is we take a look at, is energy required? So a passive process requires no ATP, no energy. The cell doesn't have to do anything, and this movement of a particular substance will occur. So what follows then are passive processes. And so you should know which processes are passive and which processes are active. So the first one that we have is diffusion. And just as a reminder, this right here, those brackets, is universal shorthand for concentration. So diffusion is just movement of a substance from an area of higher concentration to an area of lower concentration. We can shorten that and just say diffusion is when a substance moves or goes down its concentration gradient. So if you think about it, this is due to the random motion of particles. If I just spray some perfume right in that area, right where I just sprayed that perfume, those perfume molecules are moving about randomly. And it is just more likely that they're going to go from an area of higher concentration to an area of lower concentration. This process stops when equilibrium is reached, when there is no more concentration gradient. So if we're talking about a cell, and we have this cell here, and we have this phospholipid bilayer, and again, keep in mind this phospholipid bilayer, in the middle is this hydrophobic core, nonpolar. So what are the substances that can diffuse in and out of a cell? We're really going to be talking about lipid-soluble nonpolar substances. Those are the types of substances that would be able to go through diffusion if we're talking about diffusing into a cell or diffusing out of a cell. All right, so that's diffusion. Now we have a special type of diffusion, and this is called facilitated diffusion. Movement of a substance, and this one is through a channel protein from an area of higher concentration to an area of lower concentration. So facilitated diffusion, if we have our phospholipid bilayer, our membrane, we're going to have some proteins embedded, and just pretend this isn't here. So now we have this opening. So now something like sodium would be able to enter the cell. And so initially you may think that this is an active process because the cell has to build a protein and put it in, kind of like putting in a door. But this is still a passive process. And the reason is because once the cell puts in these proteins, puts in this doorway, then the cell doesn't have to do anything. So we take a look at, is energy required each and every single time a substance moves through? So what are the things, the characteristics of substances that would go through facilitated diffusion? We're talking about ions because they have a charge, so they wouldn't be able to get through this nonpolar, this hydrophobic area. And then we have amino acids and glucose. Glucose is just too large. So we can kind of look at it this way. Facilitated diffusion, those substances that will go through facilitated diffusion are the ones that have a charge and are large. So glucose is just simply because it's too big. So it needs a doorway. Amino acids kind of fit both of these categories. They're going to have a charge and they are pretty big. So that is going to be facilitated diffusion. And we can take a look here at this next slide. So here, let's just pretend this is glucose, so it's lipid insoluble. So it can't just simply go right through. We're going to need some sort of transport protein to get this into the cell, or vice versa, to get this out of the cell. And if this was sodium, it would have to go through this channel protein. And so both of these are going to be facilitated diffusion. Whereas if we have something here that is going to be lipid soluble and not polar, it can go through this hydrophobic core no problem. So that will just go through simple diffusion. So now we're going to take a look at a special type of diffusion, and this is osmosis. It's diffusion of water, just water, across a selectively permeable membrane. And this might seem a little bit backwards, but what we're going to do is we are going to be taking a look. If we have solution A and solution B, we're going to take a look at the solute concentrations to see which way the water is going to go. So we're using the solute concentrations to see which way the solvent will move. So when we compare solute concentrations between two solutions, that is tonicity. And so when we do that, there's going to be three possibilities. A is going to be equal to B, A is greater than B, A is less than B. So isotonic is when there are equal solute concentrations. So where can I draw this? So if I have 10% X inside, 10% X outside, that's isotonic. Water will still cross the membrane, and that's really important. Water will still cross the membrane. That is not osmosis. Some water will enter, some water will leave, and they will cancel each other out. And this is just because of the random motion of these molecules. So water will still cross the membrane in an isotonic situation, but that is not osmosis because there's no concentration gradient. Hypertonic and hypotonic. Now the book goes over this a little bit differently. All I'm going to do is I'm just going to say, in order for us to say is something higher or if something is lower, we needed a reference point. Right? If I say A is greater than B, it's no different than saying B is less than A. So let's say we have this cell. We have 10% inside, 15% X outside. So now the question becomes, how will I phrase the question? If I say what is the inside of the cell with respect to the inside of the cell is hypotonic. If I say what type of solution has the cell been placed in, the cell has been placed in a hypertonic, right? Because outside is greater. So you just have to make sure that you read the question to know what my reference point is. Now, regardless of how I phrase the question, in this situation, water will only go in one direction, and water will end up leaving this cell. You might be thinking, wait a minute, why isn't it entering? Because we have 15 here and 10 here. Keep in mind, those percentages are the solute. So the water, we have a lower concentration of water out here than we do in here. Okay, so another way to think of this, where can I write this? I'm going to try up here. I don't know how well this is going to work. Water will always go towards the hypertonic solution. All right, and you should probably jot that down. So, two other terms. When water enters a cell, what can end up happening is lysis. Your book might talk about hemolysis. That hemo is just referring to red blood cells. Lysis is just more general. And when water leaves, that is referred to as crenation. Okay, it's quite a bit there. just one quick example. This one is going to be a long recording, but you've heard that if people are stranded on an island or on a boat, they can't drink the salt water. What ends up happening when you drink the salt water, here's your cell. What you've done is you've created a hypertonic solution outside. Right? The salt concentration outside the cell has skyrocketed. So because it's hypertonic outside, water is going to end up leaving the cell. All right. Okay, I'm going to try and go through the next few a little bit quicker. Filtration is just when there is a pressure gradient. Hydrostatic pressure is just the pressure a fluid exerts on the wall of its container. So if we're talking about hydrostatic pressure in a blood vessel, that is really the same thing as blood pressure. Okay. So, excuse me, up to this point, everything that we've covered has been passive processes. So now what we're going to do is we're going to move to active processes. So these require ATP. Okay, I don't have it. So the first one that I'm going to cover that is not on here, at least I don't think it is. And it's called active transport. I'm going to back up a few slides so that you can get the definition of active transport. So for active transport, all you have to do is use this same definition, but just change the direction. So active transport would be movement of a substance through a channel protein from an area of lower concentration to an area of higher concentration. All right, you might want to pause it here because I'm just going to move forward now. And so when it does that, we say that it goes against its concentration gradient. Okay. All right. So active transport, that is an active process. We also have vesicular transport. Vesicular transport, we're talking about things that are just so large that we couldn't put a doorway in because too much of the cell would be able to leak out. And for vesicular transport, we pay attention to the direction. So exocytosis is when we move stuff out of the cell. So from the cell to the extracellular space, so we're moving it out. Endocytosis is when the cell will take stuff in. Now, what we do for endocytosis is we take a look at the state of matter for endocytosis. So endocytosis can be then broken up into phagocytosis, which is solid. Pinocytosis is a fluid. And then there can also be receptor-mediated endocytosis. Receptor-mediated endocytosis, what we typically see is that there is a receptor, and once the ligand binds to the receptor, it triggers the engulfing of that receptor ligand complex. Okay, so that is receptor mediated endocytosis. And I have this image here showing you how this is exocytosis. You don't have to worry about snare proteins, and if you've had Bio 111, you might have also heard of SNAP proteins. But really how this works is this vesicle containing it will just end up fusing with the plasma membrane and releasing its contents. And you can also think of endocytosis as just kind of the reverse. This stuff will go into this area and it'll just pinch off and create this secretory vesicle. Okay? And then this last bit, this is just kind of a chart. You can kind of take a look at it, write it down. I would just really, this is just a summary, an incomplete summary. I would just do something like this. And instead of energy source, what I would do is I would just do maybe passive versus active, and then kind of write all of these processes and put down examples for them. Okay. Next part, we're going to start going over the organelles inside a cell. And that's it for this